Okay, we have our last speaker of the session. This is Maria Jaime, who is going to tell us about a novel 96-well system for simultaneously housing, manipulating, and feeding flies.
Good afternoon, almost afternoon, everyone. Thank you for sticking around. I know we are all hungry, but I will try to be fast, and so we get to go to lunch. Thank you, the organizers, for choosing my abstract. And it's my pleasure today to share my work. We have been working in designing a system that allows to manipulate flies and hopefully do a throughput system with it. So first, as we know, the drugs screening and sequencing technologies have evolved a lot in the last 20 years in cell culture. And we believe that if we provide the tools for a higher throughput with model organisms, we will be able to be incorporated in these screens. So with this idea in mind, we embark ourselves in trying to come up with a system that allows to screen flies in a more efficient way that is similar to cell cultures. So for the designing parameters that we use, the main idea is to reduce or eliminate the fly manipulation, use very small volumes of drugs that sometimes is a very limited factor, use liquid food to feed flies, to have the capability that the cell culture assays they have, that you can have different treatments in the same plate without in the same assay without having any issues, ideally having monitoring capabilities, and the most important thing is they have compatibility with a 96-well plate and the potential for automation. So with that in mind, we went through several steps of designing, several attempts, and we came up with the WAFFL that stands from a whole animal feeding flat. Here we have the feeding layout, where we just have one of the parts of the system, that is the feeder plate. In this, the feeder plate, we house the flies and feed the flies. And we have a regular 96-well plate where we provide the food to the flies. Then we have the harvesting layout, where we have the complete system that we designed. This system at this moment is 3D printed, where we have the receiver, the feeder plate, the transfer adapter that is a very small interface that allows the connection between the feeder plate and the receiver plate. That permits the transfer of the flies for the harvesting and for any assay that you decide to do. So here I am showing you a closer view of the feeder plate. As you see, it's a 96-well layout. You have round wells in the bottom with square wells in the top. In the bottom of the round well is kind of a cupular form, and we have 7 openings. Each opening is of 320 microns. That gives enough space for the liquid food to go through, and the flies take it with their proboscis. At the same time, inside of the well, we are providing enough space for the fly to perk while they are going through the food, towards to the food. So here is a close-up of the 3D printed piece. So we have, as you can see, here is a close-up of the base of the round wells, and we can see the 7 openings in how they look. And also we can see that the feeding in the 96-well plate is very precise, and that came out after several design changes. And we have this little conical effect, so in that way we permit that the fly food sits perfectly in the bottom of the well, reducing the tension between the feeder plate and the food plate. And we feed the flies with a very small amount of food. We are just providing 8 microliters of food per well. And that is enough to feed the fly for 24 hours. Then we have the 2 complementary parts that they are used for the harvesting of the fly, that they are the receiver plate and the transfer adapter. As I mentioned, the receiver plate is a a plate with a very narrow wells where the flies, they go in after spinning the plate. And this plate has a lip that actually fits inside of a regular depth, 1.1 milliliter depth well plate. So in that way, by centrifugation, we harvest the flies. And the transfer adapter is just an interface that goes from the square wells from the feeder plate to the round wells of the transfer adapter. So also we are working in the monitoring capabilities of the WAFFL. So the whole idea is that if we feed with certain treatments the flies, we can observe how they behave. So we are working, right now we are designing a setup where we have 24 cameras. Each camera is monitoring 4 wells at the same time. It's a Raspberry Pi camera, and that camera is connected to a small Raspberry Pi board. And that is doing the collection of the data, and then we are processing the data. So in that way, we can see how the flies, they move and how they behave inside of the well. Another point that I would like to emphasize, as you can see, is that in each well, the flies, they have plenty space. So we are not restricting the space or limiting. So they can have a pretty normal life. So that is a very important factor. Then we have, here I want to give you just a general overview of how simple it is to work with the WAFFL. We have been doing at this point several small screens that I will show you some of our results, but I want to give you an overview of our methodology. So in the WAFFL, we can use different growth stages of the flies. We can use pupa adults. Here you can see the flies in the WAFFL. We can place pupas and they close. It's very simple to get the fly feeder plate into the food, and it's very simple to switch from one treatment to the other. We just blot the excess of the leftover food, and we pretty much pass to the next treatment, and you are moving 96 well flies. Here is very easy to harvest. We just peel the silicone mat, attach the transfer adapter with the receiver plate, give a quick spin, remove the WAFFL, attach the 1.1 milliliter plate, give another quick spin, and we have the flies. From here, you add the lysis buffer that you need, or you decide to use, and you go ahead and do the assay that you want to do. You can use a lysis buffer for RNA extraction, DNA extraction, or anything. So as you can see, the WAFFL facilitates the assay with individual organisms. So here I just want to kind of give you a summary of the capabilities of the WAFFL. It's very easy to set up. We can have a different growth stages of the organism, and also can be compatible with another model organisms. We can provide different type of treatments to the flies. We can do a continuous dose feeding. We can do a bolus washout. We can do a ramp dose of the specific treatment. And the assays that you can do at the end of your experiment is as you wish. We have done RNA-seq. We have done lipid storage. We are working with the monitoring device to assess behavior. And you can also do metabolics and proteomics. So right now I'm going to talk about some of the results of some small experiments that we have done so far. The first one, I'm going to talk about the evaluation of the bolus washout. That is a work in progress. So the bolus washout, the idea is that we starve the flies, so in that way all the flies, they eat at the same time. And then we feed the flies with a liquid chemical defined food that we add a sulfurodamide B, so in that way we can quantify the content of the ingested food by the fly. And then what we do after 1 hour feeding, we place again the fly in a starving buffer, and we do samplings over time to quantify how long it takes that bolus of food to go through the gut of the fly. So here in this graph, we have in the y-axis, we have the percentage of fluorescence, and in the x-axis, we have the time. The first part is the time that we feed. We just feed for 1 hour, and then after that, we quantify how much fluorescent signal we see. As you can see, at the beginning, after 1 hour feeding, we have pretty much the same signal between males and females. And after time goes by, the passage of the bolus is faster in males than in females. And this is a very consistent result that we are seeing. So with this one, we get an idea that every feeding to go through to the gut takes us between 1 to 4 hours. Then also I'm going to show you some results that an assay that we test different diets in the flies. The purpose of this assay is to validate that the WAFFL has a potential and is comparable to the current methods to do experiments with vials. And anyways, we're using a very small amount of food and a very small amount of drug if you are doing a drug assay. So we fed the flies with a different caloric content. So what we did is we placed Oregon R flies in the WAFFL. We starved them for 6 hours, and then we fed them with chemical-defined liquid food. We evaluate males and females, one fly per well. We have 48 replicates. We have 2 caloric contents, 100 and 400 kilocalories. We fed for 24 hours. And then we macerate the whole body, and we did the lipid measurement. So the first thing that we checked was that we are able to differentiate between a fed fly and a starved fly. And as you can see, it's very evident the difference. Here in the graph, I am showing you the triglyceride content of milligrams per deciliter and the sex in the x-axis. So as you can see, there is a difference between the blue is the starved flies. They have a lower triglyceride content in their bodies compared with the fed flies with 400 calories. And this is using a tag kit assay. And then when we compare, as you can see, males, they have a lower content of triglycerides. And there is a significant difference. So we can differentiate between starved flies and fed flies, but can we differentiate between flies with a different caloric content? Yes, we can. So we starve the flies again. We fed them for 24 hours with 100-calorie food and 400-calorie food, and we measure the triglyceride deposition. And we can see the difference between the flies fed with 100-calorie food and the 400-calorie food. So I think this is a very good evidence that we can mimic or replicate results normally obtained with experiments done in vials. We are just using the WAFFL with very small volumes of food. And as a summary, well, the WAFFL is a very versatile tool, permits us to allow and do high-throughput screens with flies and other organisms, and provides an option for automation and robotic systems, and is comparable with traditional methods. I would like to thank you, Oliver Lab, Brian, for giving me the opportunity to embark in this project, Alejandra Flores for helping me to collect a lot of data, all my colleagues in the Oliver Lab, and a lot of people that helped me to make this possible, and in special the 3D Mac facility, because they assigned me a 3D printer to be able to generate my WAFFLs and being able to assess and do all my experiments. They are across the road from us, is the Walter Reed Naval Base in Bethesda, and also the Signal and Processing Instrumentation section at the NIH, since I am working with them for the monitoring device. Thank you, and I am happy to take your questions if you have any.
Hi, just a quick question. On the harvesting of the flies, obviously, how well they're macerated can affect your variability. Can you comment a little bit more on how much volume and how easy it is to macerate the flies?
We normally macerate the flies with 50 microliters of the lysis buffer and 7 glass beads, is what you asked?
Sorry, just are they macerated in the tubes or do you take them out? And so how easy is it to get the flies? Do you macerate them in the format of the WAFFL or are you taking them separate?
No, we macerate them in the 1.1 milliliter depth well plate, 96 well plate, in that plate. And it's a very small volume. We have a bead beater, and it's very easy to macerate. So everything keeps going in 96 well format. Because also we do the RNA extraction in 96 well format or the protein extraction in 96 well format. So once that we are in the plate, we go all the way in the 96 well format.
Okay, great. Let's thank all of our speakers once again, and thank you for coming to the session.